At first, it looked like a typical neurological case. A 58-year-old woman in the US began experiencing tremors, a slight unsteadiness in her gait, and some slurred speech. Maybe Parkinson's? A stroke? The kind of conditions doctors are trained to expect in aging patients. But this wasn't that. Within weeks, her symptoms spiraled. She lost control of her bladder. Her speech deteriorated into unintelligible murmurs. Her breathing became irregular. Then she fell into a coma. Soon after, she died. It was May 2025 when the case report was published in Emerging Infectious Diseases, a peer-reviewed journal run by the US Centers for Disease Control and Prevention. The diagnosis was stunning. Creutzfeldt-Jakob disease, or CJD, a rare, rapidly progressive and always fatal brain-wasting illness. But what shocked doctors most wasn't just how quickly she died. It was the fact that the seed of her disease had been planted nearly half a century earlier. According to researchers, the woman had received hormone therapy between 1971 and 1980 for panhypopituitarism, a rare condition in which the pituitary gland, the master endocrine organ, stops producing essential hormones that regulate everything from growth and sexual development to metabolism and adrenal function. In the 1970s, this was treated using human growth hormone injections extracted from deceased individuals. These cadaver-derived hormones were common and considered life-saving at the time. Thousands of patients worldwide, including children with growth deficiencies and women experiencing infertility, received these treatments. The science was seen as miraculous, a blend of compassion and biology. But behind that progress lurked a threat no one yet understood. The very act of trying to restore health had unknowingly introduced the seeds of an incurable disease. Prions are one of biology's greatest paradoxes. They aren't viruses. They aren't bacteria. They have no genetic material. They're simply proteins, but with a dark twist. When abnormally folded, a prion becomes a kind of molecular saboteur. It triggers nearby proteins to misfold in the same way, leading to a rapid buildup of toxic proteins that destroy brain cells. Normally, prions, shorthand for proteinaceous infectious particles, are found in all mammals, including humans. Their exact function is still debated, but they seem to play roles in cell signaling or metal-ion binding. The issue arises when even one of these proteins mutates or changes shape. From that moment, the cascade begins. The woman in this case had likely received a contaminated hormone injection that carried misfolded prions, a contaminant that was virtually undetectable with 1970s technology. The prions sat silently in her nervous system for decades, slowly, and then suddenly, beginning to unravel her brain from the inside out. Unlike most infections that provoke fever, inflammation, or immune response, prions remain undetected. There is no inflammation, no antibodies, no warning. They simply build silently until the collapse is complete. This phenomenon, a latency period lasting decades, is one of the most chilling features of prion diseases. It creates a disconnect between cause and effect that makes tracing the origin incredibly difficult. Imagine a pathogen that doesn't strike until 30, 40, even 50 years after infection. In the 1970s, doctors collecting pituitary glands from cadavers had no idea that even a single contaminated sample could seed an unstoppable chain of molecular destruction in a recipient. Hundreds of glands were often pooled together during extraction, meaning that just one infected donor could potentially contaminate batches used for dozens or even hundreds of patients. And there were no tests to screen these glands. No standards for prion safety. Prions weren't even a recognized concept until the 1980s, when neurologist Stanley Prusiner won the Nobel Prize for proving their existence. That means that for over a decade, a slow-burning epidemic was hiding in plain sight. By the time the first signs of trouble emerged in the early 1980s, the damage had already been done. In 1985, a formal link was made between cadaver-derived growth hormones and a growing number of CJD cases in both the US and abroad. That year, the US halted all distribution of pituitary-derived hormones. Clinics were ordered to destroy their stocks. Medical journals published urgent warnings. Lawsuits followed. The FDA rushed to approve a safer alternative, synthetic human growth hormone created through recombinant DNA technology. This was a major scientific milestone. Scientists had engineered bacteria to produce human hormones in a lab, eliminating the need for cadaveric tissue. By 1987, these genetically engineered hormones had replaced older methods worldwide. But for the estimated 8,000 Americans and tens of thousands globally who had already received the old treatment, the damage may have been irreversible. At least 30 confirmed cases of CJD have emerged in the US alone, and the true number may be higher. Some may never be diagnosed. Others may still be silently incubating the disease. And many of those affected may never connect their symptoms to something that happened in childhood, decades earlier. For these patients, the story of their illness begins long before they feel the first tremor. Buried in a medical file no one has opened in years. Prions are not just deadly, they're nearly indestructible. They survive boiling, radiation, even formaldehyde. Standard sterilization procedures like autoclaving do not reliably destroy them. Hospitals treating known prion patients must often incinerate surgical tools rather than reuse them. Once in the brain, prions cause rapidly progressing spongiform degeneration. The tissue becomes perforated with holes, and the patient's neurological function collapses. Motor skills vanish. Personality disintegrates. Memory fades. Breathing fails. In this woman's case, MRI scans revealed the characteristic cortical ribboning of CJD, where parts of the brain appear brighter than normal due to damage. Her spinal fluid tested positive for the 14-3-3 protein, a biomarker for prion disease. The final diagnosis, iatrogenic Creutzfeldt-Jakob disease, ICJD, caused directly by medical intervention. This diagnosis didn't just mark the end of her life. It reawakened a forgotten chapter of medical history. A mistake from the past now claiming its toll. What makes this story so tragic isn't just the rare nature of the disease, it's that it was preventable. The hormone treatments she received were intended to help her grow, to normalize her life. And they likely did. For decades, she lived unaware of the fatal agent that had been implanted in her by well-meaning doctors. By the time symptoms appeared, it was too late. No treatment exists for prion disease. No drug can slow it. No surgery can reverse it. Median survival is 6 months. And because of the long latency period, there may be others, patients from the 1960s, 70s, and even early 80s, whose prions haven't yet activated. Some scientists believe we could still see additional cases emerge in the coming years. The woman's case is not just a reminder of biological complexity, it's a symbol of unintended consequences, of how even life-saving science can carry hidden costs that echo across time. Research continues. Scientists are exploring immunotherapies, gene editing, and molecular chaperones that could stabilize proteins before they misfold. One technique, RT-QICT, real-time quaking-induced conversion, can now detect prion diseases earlier and more accurately than ever before. Still, no treatment has emerged that can stop the disease once symptoms begin. Efforts to silence the PRNP gene, which encodes prion protein, may offer hope for people with inherited prion mutations. But for acquired cases like ICJD, prevention remains our best strategy. Prevention is also a challenge of memory, remembering the biological risks we've once overcome so we don't stumble into them again. The woman's death isn't just a tragedy, it's a cautionary tale. How many other time bombs remain hidden in the brains of those treated decades ago? This isn't fiction. This is the quiet legacy of an era when medicine worked miracles, but didn't yet understand all its risks. And in the case of prion disease, those risks are immune to time, temperature, and treatment. This case doesn't live in the past. It lives in the still possible future. So if you found this story haunting, eye-opening, or simply unforgettable, subscribe to the channel. Because the most powerful science stories aren't just about discovery. They're about reckoning with the truths we missed, the costs we didn't see coming, and the futures that might still unfold. Stay curious, stay skeptical, stay tuned,